agreeing to give us the seminar or this webinar. So Obinna is a program lead in autoimmune inflammation disease drug discovery at the SLAMogen Limited, which is a spin-out company, biotech company from University of Aberdeen. And his main focus is on biologic drugs discovery and drug development for chronic diseases such as autoimmune inflammation and cancers. He did his BPharm in University of Benin in Nigeria, and then did MSc in Aberdeen, got a distinction, then went on to do the PhD also in Aberdeen, which was funded by the Scottish Universities Life Sciences Alliance, or it's called SOSA. Then he also did visited UNISA to do an IP law or postgraduate certificate in IP law. He's also the fellow of the UK Higher Education Academy and holds a lectureship position in the School of Medicine, or it's called IMS, and at the University of Aberdeen. He's the president of the Scottish Registered Foundation. It's called SAIS, which is focused on bridging the life sciences knowledge gap between the UK and Africa. And in 2021, he was awarded the prestigious UK RI Future Leaders Fellowship, which is basically a £2 million grant to continue his research in advancing cutting-edge drug discovery. And I think he's doing that also in SLAMogen. Obinna, take it away. Thank you.
Yeah, thank you so much, Sabelo, for the introduction. And thank you everyone for joining today. I really appreciate the invitation to speak to you all. It's indeed an honor. I mean, a very special privilege to be in the midst of my fellow African researchers in life sciences, which is always great. So I'm gonna just take it away. You've seen the title, it's too long, boring, but yeah, we'll get there. Sabelo, how do I point the option? Yeah, I got it. So if I go, just a brief outline of what I'm gonna talk about. I think Sabelo already talked about my background, but I'll just give you a bit of flavor around my academic background, how I went from being a pharmacist to what I do today, and then give you an introduction into the soloMER technology, which is the core of the talk today about our drug discovery pipeline, and how we are exploiting the advantages of a unique binding domain in the space of drug discovery. I mean, as a biotech company as well. So one of the programs I'm going to talk, there are lots of programs we do here at SLAMogen. One of the key programs is the, AntiTnf program. We have a whole lot of other programs in autoimmune inflammation, but I'm going to focus on the AntiTnf program today. And then I'll give you a bit of a flavor on some of the other pivotal work we are doing beyond autoimmune diseases. So some in oncology, and of course in COVID-19 as well. And just a bit of a disclosure, I am not an immunologist, so you get to see why. So I'm not an immunologist, but I'm really pleased speaking to you guys today as well. So my background, in 2006, I graduated as a pharmacist from the University of Benin. So that answers the question that I'm not an immunologist. So between 2006 and 2008, you had to go through this compulsory, 1-year internship, and then the second year serving your nation. So that took me between 2006 to 2008 to complete that. And in 2008, I was lucky enough to get a job at Boehringer Ingelheim in South Africa, in Randburg, actually. So worked with Boehringer Ingelheim. So this is getting industry experience. Worked with Boehringer Ingelheim for about 28 months before deciding to move to the UK to study masters, to get a master's degree in clinical pharmacology and toxicology. So completed that, and in 2012, got a scholarship to pursue a PhD in biomedical sciences, but focusing on molecular biology and protein engineering. And then again, that means I haven't done anything in immunology. Okay, so I kind of learned on the job. So one thing that happened, one of the things that was very consequential in my life was meeting a few people, most especially, people of African background doing so well in their research. So I came across a few guys doing PhDs at the time when I started my PhD. And one of the people I met was this guy, Sabelo. So, I listened to his presentation when he was still, I think, second year or thereabouts, and it blew me away. And that was an inspiration for me to try for the best in my space, to keep pursuing my dream. And Sabelo very kindly, as being a junior student to him, always invited me over to his house whenever they had a house party. So this was one of the military costume house party he had, and I was there. But the other thing about these guys, not just Sabelo, but most of the African guys I've come across, is their love, their passion for science. This was on their graduation day, and Sabelo having a chat on their graduation day discussing macrophages. Who does that? That was very interesting. And just to point out as a disclosure, the person Sabelo is talking to was my girlfriend at the time, who is my wife today. So the love for science is obvious when you see most of the African scientists here as well. Now, and of course, graduated in 2016, and luckily, just at the time I graduated, we spun out a company from the University of Aberdeen. So it's an academic spin out called the Lasmodin Limited. So this company was spun out on the back of some of the intellectual property that we have acquired in the past and also during my PhD. So those were the core IP portfolio of the company. And then because you can see now moving away, still doing core research, but more commercially oriented research. I was a bit out of my depth trying to understand the business of science. So I decided to, get an online degree in intellectual property law. So of course, my love for South Africa, I always want to go back to South Africa. So I chose UNISA for the online degree, and I got a little bit of funding from the World Intellectual Property Organization to pursue that postgraduate certificate course. And of course, in 2020, the pandemic happened. And I'm going to tell you a little story how we contributed to the fight against COVID-19. And in 2021, I was awarded a fellowship of the Future Leaders Fellowship Program, UKRI. So I got the £2 million funding to keep supporting to continue developing my science, and at the same time develop my leadership skills as well. So currently building a team of scientists that will work with me in the next 4 years on this fellowship. And as Sabelo also mentioned, and I hold fellowship at the Higher Education Academy because I have a huge teaching commitment at the University of Aberdeen as well. So I do teach, from medical science courses up to the business of science as well, to undergraduate and postgraduate students as well. So just a little bit of a news, which will be in the press soon. So Lasmodin is just closing, a round of funding at the moment. So that should be in the press probably by the end of today or tomorrow. So just keep an eye out for that announcement. So where are we actually? So the company is located on the health campus at the, just around the University of Aberdeen Health Campus as well. So this is where we are. And this is the University of Aberdeen School of Medicine, Institute of Medical Sciences. And if you go down here, this is the Aberdeen Royal Infirmary. So that's the teaching hospital. So we call it the Triple Helix Network. So that we are in close proximity with each other. So the biotech commercial entity with the academic and the clinicians always partnering and staying in close proximity, which is very essential. So, before I go into the science, I always want to share my philosophy when it comes to research and innovation. I'm a big believer of science. I'm a big believer of commercializing our research and innovation. The only reason, one of the main reasons why I think we should strive to commercialize our research and innovation is because it does not just only create jobs, but it creates better science. It creates bigger hardware and creates better science. It allows us to build, cross-sector, cross-disciplinary partnerships. And at the same time, the academic research hub is very, very essential in all of this because the brain comes from the academic institutions. But it is essential that we continue to think about commercializing our science. That limits our dependence on the university funding, limits our dependence on government funding, because you are developing your science, you are going out there looking for investment, you are creating jobs, you are adding impact to the society. And at the same time, you are bringing in new expertise to your field, to your society as well. The reason why I say this is I look at the global pandemic, which all of us witnessed. And it scares me to see that in Africa as a whole, we constitute about 16% of the global population, but we contributed almost nothing to the entire innovation. In terms of drug discovery, vaccine discovery, we contributed nothing, and it is scary. It is very scary that, if this was a very deadly virus, you can't tell the UK government not to look after their people first. These countries, developed countries, will look after their people first. We could have all been wiped out. So we need to step up our game. We need to really step up our game. Try to find, because the moment we start commercializing, things will change. There will be money coming in. There will be new expertise coming into the science sector in our country. And I say this because you look at the patent applications. I'm looking at just a snapshot between 2010 and 2020. Exactly what I'm saying. In the entire science and technology sector in Africa, we in 2020 only contributed 0.5% of patent applications. And when I say 0.5%, majority of the 0.5% were actually Africans residing outside Africa. So these are non-resident patent applications. So as I said, we just need to step up our game. But yeah, so back to science. So what I'm going to talk about is just to show you about, first of all, introduce you to the technology, what we do. And I say the smaller, the better. We can argue about that later. So the classical monoclonal antibodies, as we all know, it's a 150 kilodalton molecular weight protein. It's usually referred to as a magic bullet. Of course, they are very specific to their target, but we look at this classical molecule. We are talking about, multiple chains, complex post-translational modifications, around these classical molecules. But this molecule has been successful, since the first monoclonal antibody was approved in the mid-1970s. Up till today, we have over 100 FDA-approved monoclonal antibodies in the clinic. So they've been hugely successful, both in oncology, in immunological diseases, in diagnostics. They've been hugely successful. However, one thing scientists, researchers in antibody space do know, what we have come to understand, is that some of the complications that come with using classical monoclonal antibody as a therapy, most of the complications are associated with the structure, the complexity of the structure. The other problem is the cost of manufacturing these products. It's quite huge because you have to manufacture these products in a highly specialized mammalian expression system, which is costly as well. So what scientists are doing is we start adopting what we call the reductionist theory. So going from 150 kilodalton molecule and start fragmenting this antibody. So what you end up with, so trying to reduce the size, because we know size, by reducing the size, you reduce the complexity, you make it simpler. So going from 150 kilodalton protein to, up to 25 kilodalton as a single chain FV, trying to break down the structure. And also by doing that, you're able to then have flexibility to create what you call the bispecific molecules around this, because a classical monoclonal antibody is monospecific, although bivalent, but monospecific. So this reductionist theory has been ongoing, and to date, it's been successful to an extent, right? So we have over 200, peptides, antibody fragments, and classical antibody approved for use in the clinic. Now, the interesting thing is, we are doing this reductionist theory today. However, in the shark immune system, this reductionist theory has already been solved 450 million years ago. So in the shark immune system, we have this ancient immunoglobulin family called the IgNAR, so immunoglobulin new antigen receptor. So this is the IgNAR structure. But one thing we know is that if you look at the binding domain, it is just a single heavy chain. It has no light chain pairing like you have with a classical antibody. So it's just a single linear chain that is actually binds to its target independent of the constant region as well. So this is a 12 kilodalton protein, between 11 and 12 kilodalton. So you can see that reductionist theory already happened 450 million years ago. And by looking at so that's binding domain, we call it the VNAR, we call it soloMER, depending on the context. But the VNAR, you can see how the structure is diverse. So the binding loops are very diverse. And I'm just pointing out the three isotypes. We have more than 3 isotypes of the VNAR. But you begin to see the binding loops, how they can be constrained, how they can be protruding with a disulfide bridge to keep them in place. And also you see how they can as well be flexible. So this binding loop flexibility, the diversity you find in there means that, and of course, as I said, that's what we call the soloMERs. But the interesting thing is because of the way the loop is different from the classical antibody, they can bind epitopes on their targets differently as well, not just the size. So when we talk about the size, there are huge benefits for reducing the size or for starting with a binding domain that is small in size. So one of the benefits, of course, of size is that you can penetrate tissues, you can target intracellular molecules, you can, use smaller size domain to cross biological membrane. So if you talk about this in the context of a solid tumor, you want to have a drug that could, penetrate the solid tumor environment, the microenvironment, get into the microenvironment. So size is very important here. And when we talk about shape, binding to novel epitopes that are otherwise not accessible to classical antibodies, it's very consequential. And it's, I'm going to show you data to support that later, why it's important that the soloMERs are binding to epitopes that are recessed, cryptic epitopes on target. And, the other thing is the stability. As I mentioned earlier, these domains have no multiple chains, so they're just single chain only. What that means is that there's no falling apart of chains, and it just requires the ability to refold and retain its native structure. That gives us the flexibility to think about administering these drugs because of the size, the stability in multiple different ways. We can store them at even room temperature at 100 degrees. You can boil them and they retain back to their structures. These are data that we have already published, in support of the stability of these domains. And the stability allows you to administer them through multiple routes. Now, because they are simple, I talked about the cost of manufacturing of classical antibody. These domains, the only post-translational modification involved here is disulfide bridge formation, which means that you could actually achieve the disulfide bridge formation by expressing these domains in E. coli. You can go as high as expressing in Pichia in yeast. And so that means that the cost of production of this end product is actually significantly lower than the cost of production of a classical monoclonal antibody. And because they are single chain, we can then begin to think of creating multivalent, multi-specific, multi-paratopic domains, so you can begin to then build them up like as a Lego, rather than taking something that occurs in nature as 150 kilodalton and start splitting them. In this case, it is 12 kilodalton, you can begin to build them up. So we use molecular, antibody engineering skills. We could create, a linear trimeric construct like this. And we could as well create, depending on the secondary mechanism of action we are looking for, we could also as well fuse the soloMER to the N terminal of or C terminal of an FC domain. So IgG1 human, FC, if we are looking for an effector function. So we could do this. We could also create what we call the quad construct. What that means is that we have 4 binding domains coming off the human IgG1 FC. So you have that flexibility to have multiple binding domains on an FC entity. So as a company, drug discovery company, we have programs, internal programs in autoimmune drug discovery. We have programs in oncology. We also do lots of partnering as well. So co-development, one of our co-development program in oncology is actually at just finishing the late stage preclinical. So we're just waiting for the data to be fully analyzed, the safety and toxic data. And our programs, other programs in autoimmune inflammation is also approaching the late stage preclinical development as well. So the first program I'm going to talk about is the Tnf Alpha program. So basically, Tnf Alpha, of course, it's discovered in the late 70s as an anti-tumor biomolecule. Of course, that never materialized. But sometime in 1985, it was found to be secreted by macrophages, okay? And the scientists actually realized that it was actually Tnf alpha was responsible for the severe, body weight loss, muscle loss that were being seen in parasite-infested animals. And of course, since then, we now know that Tnf alpha, being a pleiotropic cytokine, does play key roles both in health and in disease. So it depends on the context we look at it from. Tnf alpha may be a good player, and in other contexts, a very, very bad actor. So I just put this diagram just to show how Tnf alpha is in the center of most chronic autoimmune or inflammatory diseases. And you can see both in Crohn's disease, rheumatoid arthritis, uveitis, and psoriasis, it plays key role in all of this. And I'm not going to dwell on the signaling, but I need to point out something very important here is that you look at the Tnf molecule. So the Tnf molecule comes as a precursor form, which is the transmembrane Tnf alpha, expressed as a surface type 2 polypeptide, which is about 26 kilodalton in size. It's expressed on activated macrophages and also lymphocytes as well as other cells. But basically what happens is that this transmembrane TNF alpha is cleaved by a metalloproteinase called the TNF alpha converting enzyme into the soluble form. So you can actually have the transmembrane TNF alpha acting, as a receptor or as a target on the cells expressing it. And you can also have the soluble TNF alpha, in the blood, binding to receptors. So soluble TNF alpha binding to TNF receptor 1, TNF receptor 2, but preferentially the TNF receptor 2 would always bind to cells expressing the membrane TNF alpha, would bind to the membrane TNF alpha. But the downstream effect is context dependent. So it is not a one-way high street. As I said earlier, TNF alpha plays role in health and disease. But what I'm just pointing out here is that, the transcription factors that could be activated, phosphorylated to lead to the transcription of genes that code for pro-inflammatory cytokines. So you end up having, in a disease state, you end up, if the trigger here is an inflammatory trigger, you end up with expansion of, further production of pro-inflammatory cytokines by the activation of these transcription factors. There is also a reverse signaling that occurs. So what we call outside to inside signaling that occurs when TNF receptor 2 binds to membrane TNF alpha. So basically, what we are trying to achieve here in terms of mechanism of action is to neutralize, first of all, to bind to TNF alpha and neutralize it. So in this diagram, I'm showing you the top 5 anti-TNF antibodies in the clinic. So these drugs have been in the clinic for the better part of 17 to 20, 23 years. So, and they've been hugely successful. The mechanisms range, and you can see there are different structures. So I'm not going to go into details of that, but basically what you see is that first in the mechanism of action is the ability for these anti-TNF domains to neutralize TNF alpha, whether they are neutralizing the soluble form or they are neutralizing the transmembrane form of TNF alpha. Okay, and the second part is the outside to inside signaling. This is very important, especially say, again, it's context dependent in Crohn's disease, where you have a lot of in Crohn's disease, where the disease is it's driven differently, where you don't just only achieve healing by neutralizing soluble TNF alpha. You need to actually induce apoptosis of the lamina propria T cells, so induce cytotoxicity of this, or apoptosis. So that means it is very essential that you do not only bind to the transmembrane, but activate the outside to inside signaling that occurs. So, and also, of course, by doing that, you could release some anti-inflammatory cytokines as well. So the other part is, but this top mechanism, which is Fc-mediated apoptosis, can only be achieved if your modality has an Fc domain. So you can see that is only achieved with these domains, but not with this, okay? So the Fc, meaning that the Fc part of your anti-TNF is binding to either the Fc gamma receptor or it's activating complements. And of course, the anti-TNF part is binding to the transmembrane TNF alpha. So at the end, in the end, you are achieving cell lysis, which is beneficial, as I say, depending on the disease you are targeting. But all in all, that's mostly the mechanisms that you see with anti-TNF modalities. And in terms of whether TNF alpha is at the center of inflammation, you can actually see that from the success of these modalities in the clinic. You have, as I said, infliximab was approved for using since 1997. It's got about 8 or 9 indications. You have Humira, which was approved in 2003, about 11, 12 indications. And in orange, you have 5 anti-TNF modalities. These 5 drugs sell over $85 billion every year in sales alone. So they are hugely successful. And then you have other, pro-inflammatory, modalities coming into play, the anti-IL-23, the JAK inhibitors, and all of that. But the anti-TNF still retain the major share in the market as well. So your question, if you're asking a question, why another anti-TNF? Absolutely, you're right for asking that. But I then have to tell you that the anti-TNF biologics that you see, they do have the highest level of warning label by the FDA and other regulatory authorities. So this label called the black box warning, because of the life-threatening adverse events that this class of biologics actually cause. So we know that the adverse events, which are life-threatening, goes from life-threatening infection to, you could develop cancers, multiple different types of cancers by taking the anti-TNF, because essentially you end up becoming immunocompromised by taking the anti-TNF therapy, although they have been successful, as I said. So we now know that those adverse events are mechanism, class dependent. So the mechanism of action of the anti-TNFs are driving those adverse events. And the other thing is immunogenicity. We talked about the complexity of the monoclonal antibody. So there is a need to solve some of these problems. And the other thing is that 30% of patients do not actually respond to anti-TNF therapy, or they do fail to maintain a response after initially responding, okay? So some of these patients, we could class them as primary non-responders or secondary non-responders. So if you look at a patient who has been, if you look at a cohort of patients who started taking TNF alpha, you will see that in 1 year, only 65% of them will still remain on an anti-TNF alpha therapy, because the other ones stopped responding or they stopped because of adverse events. Then if you look at that for 5 years, only 40% remain on an anti-TNF therapy, meaning 60% became either non-responsive or they just stopped because of life-threatening adverse events. We now realize the market for that refractory population is growing, and it's estimated to reach $3.6 billion by 2024. So, these are the little incentives driving, new modalities in anti-TNF space. So basically what I have done, or what I have done with my team, is to create this quad construct by, of course, identifying, the VNRs that bind differently to the TNF molecule. But by creating this quad construct and also a smaller linear construct. So you're looking at 100 kilodalton, 45 kilodalton as compared to 150 kilodalton. So here we are looking at the gold standard, Humira, which is the best-selling anti-TNF drug. But by creating this alone, I have improved the in vitro efficacy of this domain by at least, in order of magnitude compared to Humira. So picomolar, 2 picomolar compared to 30 picomolar. And this linear construct seems to be comparable with Humira already, but remember, just 45 kilodalton in size. But the other important thing is looking at the immunogenic epitope. So I have now significantly reduced the immunogenic epitope from 21 to 10 by in silico analysis, using in silico methods to assess that. And of course, the immunogenic epitope dropped to 6 as well for the linear construct. Now, one other thing I needed to validate was I needed to show that the binding profile needs to be different. Remember, I've shown you the structures of the VNRs, but I need to also demonstrate that the binding profile is different to Humira, for instance. Humira, when I say Humira, I mean adalimumab. Humira is just a brand name. So, and you can see this is looking at binding to TNF alpha and neutralizing TNF alpha that comes from different species. Okay, all 3 domains here, this is a single domain, so single domain from camel. You are looking at them, they all bind and neutralize human, dog, cyno, TNF alpha. But the moment you start looking at mouse, you have Humira doing that, except not these 2. And the moment you check pig, you see, these 2 doing, just binding differently. So there is already a distinction in their binding profile already. So the only way to confirm everything I've just told you is to do a co-crystallization work. I mean, it costs a lot of money. It could take years, it could actually take weeks, and it could take days to solve the crystal structure. So, looking at the structure, this is the crystal structure of TNF. TNF occurs as a homotrimer, as you all know. So it comes together as 3 TNF molecules. So you have the TNF homotrimer here, and in blue ribbon, you are going to see the soloMER where it binds to the TNF molecule. Okay, so this is a co-crystallized work. We're showing you where one of the soloMERs bind to the TNF molecule. And then if we then pull all the existing anti-TNF modalities, all the anti-TNF drugs in the clinic, and try to look at the footprint, because we have solved all these crystal structures have been solved. You put the footprint on a TNF trimer molecule, you will see that the footprints, this is exactly in red where they all bind on this molecule. Okay, and if I overlap that with the soloMER binding footprint, you see how different. So soloMER binding footprint is in blue. You see how different it is from all the existing anti-TNF molecules. So the hypothesis is that by binding to this unique epitope, we are able to drive a novel mechanism of neutralization. So that is something we are now working to validate, and that paper will be out very soon. So one other thing we've done, of course, was to show a proof of concept efficacy in an in vivo disease model. So there are a few disease models we've done, but for the benefit of time, I'm just going to share one, and then, there are a lot of papers that we've published that talks about that disease model. So here you are looking at a transgenic mouse model of polyarthritis. So this mice developed polyarthritis at week 3, and the disease is 100% penetrance, which means every single mice with that genetic modification will develop polyarthritis at week 3. Okay, so, and when we're able to show using the superpotent quad construct and the linear construct as well, but with the superpotent quad construct, we were able to demonstrate that even as low as 0.5 mg per kg dosing, we were able to completely block this infection or block polyarthritis in these mice. But compare that to Humira, so the Codex blocking at 0.5 mg per kg and looking at Humira with disease breakthrough at 1 mg per kg, and even at 10 mg per kg Humira, you still find some disease breakthrough as well. But looking at the histopathology score, you can see the Codex, at 1 mg, just doing head-to-head comparison of 1 mg per kg Humira and 1 mg per kg Codex, you can see the significant difference in histopathology scores. So the other important thing is immunogenicity. What I've told you earlier, we could use a silico model to determine the immunogenic epitopes. But of course, in the longer run, you want to do real-life dendritic assays to actually quantify this antigenicity. But using different models of in silico analysis. So in this one, we are using EpiVax, which is a validated in silico model. And you see the quad domain, coming off here as significantly less antigenic than Humira or Remicade, which is Remicade is infliximab. Okay, so that kind of validates everything I've said. And the other thing, which I won't have time to talk about today, is the anti-drug antibody, which is a huge thing in the use of monoclonal antibodies as therapy. So the neutralizing anti-drug antibody would always neutralize your drug and aggregate with your drug, and your drug is rapidly excreted from the patient. So that leads to loss of response, loss of efficacy. So, and it's a huge problem with monoclonal antibodies. So in this paper, I was able to demonstrate that the quad construct actually do not elicit neutralizing anti-drug antibodies as well, compared to Humira. So that's the TNF work. It's quite a lot of things going on, but just to give you a flavor of everything. Okay, so just going to talk a little bit about our work in oncology. So, and this is where here we are targeting the ROR1. So ROR1 is actually an orphan receptor tyrosine kinase like surface antigen, which is expressed by many tissues during embryogenesis, but with limited expression in adults. Okay, so once we mature, ROR1 expression is very, very limited. But we now know that ROR1 is overexpressed in multiple cancers. Okay, and it is now associated with poor prognosis. So poor prognosis in triple-negative breast cancer, lung, ovarian, colorectal cancers. So what I'm showing here is the structure of the ROR1, a classical ROR1 structure, and I'm showing you 3 lead candidates that are soloMER candidates, and where they bind. There is this diversity and flavor where you bind to. So we're able to identify 3 soloMERs that bind different epitopes on ROR1 molecule. But then again, I've talked about magic bullets. The classical monoclonal antibody or any binding domain is you could call them magic bullets, because you select them against a specific target, they would act like a homing device and selectively bind to that target. But in oncology, we now know that, of course, cancers are stochastic. Cancers do evade mechanisms. They do evade response to drugs. They develop resistance to a particular therapy because they can have an escape mechanism where they continue to propagate. So it then became essential that you no longer rely on monotherapy, or you no longer rely on just targeting one overexpressed receptor. So in this case, the antibody drug conjugate is a concept where you have your antibody that binds to a specific target, one or two specific targets that is overexpressed on a, say, solid tumor, on the surface of a solid tumor. But then you, using clever chemistry, you conjugate a very cytotoxic payload to this drug. So this payload could be, DNA damaging, it could be topoisomerase inhibitor, it could be, tubulin polymerization inhibitor. So, but the point is to conjugate just the right amount of cytotoxic payload to an antibody so that it only specifically gets to the site of tumor and releases the payload. You don't want it to release the payload, around healthy tissues, because the cytotoxic payload is non-discriminatory. But with the help of an antibody or a binding domain, you can easily direct that to the tumor sites. So that's exactly the one we adopted. Of course, you see what we have done here. We have the soloMERs against the ROR1 fused to a human IgG Fc domain. But of course, using our proprietary linker and then using the cytotoxic payload. Here we have used the PNU anthracycline, of course, which is a DNA damaging payload. But this is a work we are doing. It's a co-development work with Armac Drug Discovery. So Armac Discovery is a pharmaceutical company as well. So they have expertise in chemistry. So you can see that's exactly the modality that we have used here. But of course, in the first instance, you need to validate that this antibody structure does actually bind to ROR1 expressing cells. And in this case, as I said, ROR1 is expressed in adulthood, but it is limited. So what I'm saying in essence is that healthy tissues, healthy cells might still have ROR1, but not overexpressed. So we need to have this domain, we need to demonstrate that this domain discriminates between high ROR1 and low ROR1 expressing cells. And here you're looking at A549 is a human lung carcinoma that is high expressor of ROR1, and the A427 is low expressor human lung carcinoma as well. And you can clearly see that we are seeing a high KD-apparent, very potent KD-apparent in terms of binding affinity to high expressing A549 or one expressing A549 cell line. And here we try to show the potent killing, targeted mediated, killing of ovarian teratocarcinoma. So in one case, in the black, we have the ovarian teratocarcinoma with ROR1, and we have knocked out ROR1 from this ovarian carcinoma. And you see there is no non-specific killing here. So the only killing that we're seeing is with the high expressing ovarian teratocarcinoma cell line. So of course, the proof of concept here relies on us, taking this in vivo to demonstrate truly that this actually can lead to complete remission of tumors. So this is a PDX tumor, so patient-derived xenograft tumors. Of course, these are all one positive patient-derived xenografts. And what we try to do here is, of course, trying to do dose finding in preclinical mouse model. And in this case, we've only done one dosing schedule, but at high dosing, so 0.3 mg per kg. We dosed at day 2. Okay, and out of the 5 mice in the group, 4 out of 5 of them, were in complete remission, over this period of time with just one dosing schedule at 0.3 mg per kg. However, dosing at 0.1 or 0.3 mg per kg, dosing 3 times, so day 2, day 6, and day 10, we see that when we dose at 0.3 mg per kg again, but with 3 dosing schedules, 5 out of the 5 mice in the group were in complete remission, whereas a lower dose of 0.1 mg per kg with 3 dosing schedules, only 2 out of the 5 were in complete remission. So that kind of begins to tell you what the dosing range and should be like. And then, so in this case, we have used 0.3 mg per kg, of course, and a more extended study. But here we have dosed 5 times, so day 2, 5, 8, 12, and 15. And up until day 105, these mice remained in complete remission from the tumor. So this work has now gone on to complete, preclinical talks, and we're just waiting for the data package to be analyzed. So I guess the next phase of this will be, clinical trials. So this is, as I said earlier, it's just completed late stage preclinical development. Now, finally, just I do have some time, of course. So just to talk about SARS-CoV-2, the COVID-19 pandemic. So basically, we're not an anti-infectious company, but of course, we have the expertise to discover therapies against anything, as long as there is something to target there. So at the start of the pandemic, that was just first week in April of 2020, we got this Scottish funding to immediately expedite drug discovery for COVID-19. Of course, at that time, we didn't know whether it was going to be a vaccine, it was going to be a therapy that was going to work. Nobody knew. So, of course, with the experience researchers have had with beta coronaviruses with regards to vaccine, it wasn't looking good, so there was, we were all firing at all cylinders, trying to find the best approach. So immediately, I think within that first week, I returned to the lab, we were all in lockdown. So returned back to the lab, got a few members of the team back, and we started trying to isolate soloMERs against the SARS-CoV-2 receptor binding domain. So we are focused on finding something that will neutralize or block the receptor binding domain, because there are already publications. I mean, with the SARS-CoV-1, we already know the mechanism through which the SARS-CoV-2 gets to infect the host. So this paper we've published in Nature Communications just at the end of last year. So just detailing everything with regards to the work. And again, I need to mention everything with regards to SARS-CoV-2 research we did is in the public domain. We are not intending to commercialize it. So the sequences of all the lead candidates that we isolated, we've characterized, they're already published in the paper. So people can go ahead and develop it as they wish and work with it and do research as they wish. So we have no intention to commercialize this. So we all know how this mechanism works. So I'm not going to tell you about the binding to host cells, but the concept here is to have the monoclonal antibody try to block this interaction between the S1 RBD and the host cell. So in this case, will be the angiotensin converting enzyme 2. It could be the aminopeptidase, it could be the dipeptidylpeptidase, the DPP4, as with regards to MERS viruses. But in the context of this, we just want to bind and block this interaction. By doing this, we prevent the fusion and entry of the SARS-CoV-2 viral particle. So in the first instance, we were able to do all of this in the lab, and all the data, it's in that Nature paper. So we were able to do this just in vitro. We initially got about 24 kits. So we had 24 binders to the RBD, something around 24 thereabout. But of course, as we started characterizing, the numbers started dropping off. And when we started going into pseudovirus studies, in vitro pseudovirus blockade, and looking at replicon competent SARS-CoV-2 neutralization, we arrived at these two candidates called the 3B4 and 2CO2. We also observed that 2CO2 was consistently superior with regards to neutralizing potency over 3B4 in all the in vitro models that we tried. So when we solve the crystal structure of both the 3B4 with the SARS-CoV-2 RBD and the 2CO2 with SARS-CoV-2 RBD, one thing was apparent. So as you can see in this structure, this is just superimposing both, and you're seeing that the 3B4, the soloMER 3B4 and the 2CO2 bind on the opposite sides of the RBD, validating everything. So they are very diverse in their interaction with targets. So they bind to different parts, which of course alludes to the fact that they will neutralize SARS-CoV-2 RBD using distinct mechanism of actions. So one of the mechanism of actions, so for the 3B4, we know that for spike protein, the RBD to interact with the S2, the RBD needs to be in the up conformation, okay? That up conformation is quite unstable. So SARS-CoV-2 RBD will not prefer to remain in the up conformation except it is interacting with the S2. So that up conformation becomes vital for binding to S2. So with 3B4, and the other thing I need to mention is that the 3B4 and 2CO2 do not bind anywhere close to the binding site of S2. So neither of these directly competes S2 off the RBD. So there is no overlapping, epitope between these soloMERs and S2. So the S2 binding site on the RBD is still intact. But what 3B4 does is that 3B4 locks the RBD in the up conformation, which is the unstable conformation that it doesn't want to be in, except it is binding to S2. So it locks it in that conformation, but while locking it in that conformation, S2 tries to bind to RBD, which is in the up conformation, but then there is a steric hindrance. So there is a clash between 3B4 and S2. And it is that clash that takes the S2 off the binding side. So it's not a direct blockade, but it's a steric hindrance method of neutralization, which is pretty cool to see. But then when you look at the 2CO2, 2CO2 binds both the up and down conformation of the RBD. So, and you can see here, I'm just showing you, this is 2CO2 binding on this, side where it binds on the RBD. So when we superimpose this, you see the S2 comfortably binding to its epitope on the RBD. But basically what happens here is that, remember, if the up conformation is not available, S2 cannot bind. So what 2CO2 does is that 2CO2 most likely binds, because it binds both up and down conformation, most likely binds most of the down conformation and makes the up conformation variants to be few, like you have fewer up conformation variants available to S2. So in most cases, you have most of the RBDs in the down conformation blocked in the down conformation, and they don't get to be in the up conformation. But we believe that that mechanism is not sufficient enough to make it more potent than 3B4. So one thing we are thinking, which we need to structurally validate, is that we are beginning to see that it is possible that the 2CO2, while binding the RBD in the down conformation, also pins down the N-terminal domains, which is, this is now looking at protomer 1, which is one of the RBD, and then another N-terminal domain. But it pins both of them down, which means that you do not get the up conformation at all. So it's not a situation where you have maybe 10% up conformation. In this case, it pins them down that you don't actually get the up conformation. This we haven't validated. We are hoping to structurally validate that. So just to summarize, just look at the two, there are a few future directions for biologics. And one of those is what we are now looking into, because of course, you look at all the classical monoclonal antibodies, they are all given by injections, right? So there is also another layer of adverse events that come by the injection as well, and, the discomfort for patients and the cost of that procedure of having to go to the hospital to get an injection or, manufacturing injectables. So it's to try and create biologics in an orally deliverable form. So what we are trying to achieve here is to deliver these soloMERs orally. It's a high-risk program, but of course you understand why it's high risk. You're talking about the low pH in the gut, you are talking about the gastric enzyme, like the pepsins and co, the small intestinal peptidases and co. So trying to achieve that is actually the next thing in the biologic drug discovery space. There are lots of companies already working on this, and there are products already making it into the clinic. So it's a validated process. So that's one way. And the second thing that people are looking into as well is GPCRs and ion channels. We now know that over 270 GPCRs and ion channels are actually implicated in a number of diseases, chronic diseases. So it's a very exciting space. So of course, it's been very difficult in the past few decades to find antibodies to these ion channels, just simply because we didn't have enough knowledge on how to produce these in the lab in enough quantity to actually carry out the selection process. And that is one problem. Second problem is that GPCRs, ion channels need to be in an active state for you to actually be able to identify something that is a relevant binder or neutralizer or blocker. So if you express these GPCRs and you're expressing them in an inactive state, then it's just a waste of time. The other problem we had at the time was, trying to get rid of impurities when you express these, GPCRs or ion channels was quite difficult. But, to date, I think about 2 or 3, antibodies in the clinic targeting GPCRs. And there is a lot of work, a lot of interest, because now people are getting a better hang on how to express these domains and to use them to do drug discovery work. So in summary, I've shown you how the soloMER platform actually does, interact with targets and, the whole discovery process. You've seen how robust they are, against specific targets. I've shown you a few data to support their preclinical efficacy, and of course, how translatable these domains are. We have both in-house, as I showed you earlier, internal programs, which is quite huge. I haven't updated everything on the slide. And also we do lots of collaborative programs. Mostly when I say collaborative, we do collaborative programs with academic institutions. We tend to also do co-development programs as well with biotech, pharma companies as well. I guess I will stop there. Thank you very much for listening, and I will take your questions if you have any. Thank you.
Thank you, Obinna. That was fascinating. It's really fascinating how far it has come and what you guys have been able to achieve. It's incredible, really.
Yeah, thanks.
And just all the things that you're thinking about ahead in terms of drug discovery and where the potential pitfalls are. Yeah, I think we probably in many countries are quite far behind. I'm just checking if we have specific questions. Okay, right. But I think for me, I'm sort of still lost as to how do you actually get to a point of selecting a hit? I mean, where does it come from? I guess maybe that's the question.
Absolutely. So that's a very valid question. So the thing is, because of the time, it's for time of the talk. I wanted to just show you guys just a few of what we do. The selection process that happens before all of this, it's a whole world of its own. So basically, what happens is there are different approaches to this. One approach is that we can take the target of interest and immunize sharks with the target of interest. Okay, So think about it this way. These sharks evolved 450 million years ago. So they are totally divergent from, our human lineage. What that means is that everything we see in humans are very new to sharks. So they would have immunogenic response. So that is the best approach for us to immunize sharks with, say, the Tnf Alpha, for instance. That program was through, so the hits came from sharks, immunized shark blood. So when we get the blood out, we start building a library with the RNA. Okay, we get the RNA, create this cDNA, then build a library around the cDNA. Then we start what we call the phage display. So you could do phage display, you could use this display. So this display technology, the principle behind it is that you have bacteriophages that then every single vena in that library will be displayed on the bacteriophage. And then you present your target again in an in vitro setting. Only the ones that can bind will be pulled out. Okay. So the phage display technology is quite powerful. It's a powerful technology. In fact, the guys that discovered it actually won a Nobel Prize for it. So that is one way. But by immunizing sharks, you are talking about 6 months. That's the process. It takes a long time. So sharks do take a while, because I told you the IgNA is ancient, right? So the IgNA is it's part of the adaptive immune system in sharks, but it doesn't. It's one of the responses that comes later. So we have to do multiple immunization boosts before you see the IgNA titer begins to come up, right? So that's why it takes 6 months to do that. The second thing, and of course, we are very, you've been here, so you know how animal use, we have to be responsible with the use of animal. These sharks, we don't kill them, of course, we can immunize them multiple times, 10, 20 different antigens. So, but we also tend to build the vena libraries in silico, right? So we design that in silico. Remember, one shark can only give you one library, right? And one antigen at a time, right? But in the test tube, we can actually build a library that contains about 1 million sharks in a test tube.
Right.
So also what we do, we have the synthetic library where we have built the synthetic library to select against any target. So the SARS-CoV-2 program I talked about was through the synthetic library. The RAW1 program, of course, there was an immunization part, but also a part of it came from the synthetic library. So where we just take the synthetic library and go straight to the phage display selection, right? So you circumvent that 6 months of immunization, and just within 2 weeks you get your hits. If the target is again, it's we have. I can't say we are successful. We can't say that every target will be successful, right? So we need to first of all look at the target, make sure the target has, all those loops I'm talking about, all those, recessed epitopes. Then we can then present that to the venas and see what happens. Yeah.
Wow, that's actually very. I guess it's very interesting in a sense that. Because I was just thinking that if the whole program was around immunizing one shark, then obviously that means you are very limited, or either you have to have a lot of sharks, or you're quite limited in what you can do. Yeah. But it seems that you are. But is the idea to spread to other infectious diseases, or mostly to stay within cancers and autoimmune diseases?
To be honest, we are not an anti-infective disease company. Okay, we don't want to be one. We don't try to be one. But yes, we do support programs for people who want to. As you can see with the COVID program, right? We contributed our expertise, with regards to that. But it is not something we are looking to get into as a company. If you look at it from the okay. I have to say this, I'm sorry. I mean, you already know where my ideology is when it comes to research, right? I look at the money, I look at, it has to be commercially sensible, right? It has the only reason why it has to make this commercial sense is that you want to be able to get an upside so that you can reinvest in the research again, right?
Yeah.
Which is, it's very important. It has to be sustainable. So if you look at the market opportunity for anti-infectives, don't forget, antimicrobial resistance is a big global crisis, big global threat, right? But you see that investors are actually not keen on antimicrobial. It is not what they are looking for. They are looking for oncology. They are looking for those high risk, high reward programs, right? So because of that's another reason why maybe, I mean, this is my personal view. I wouldn't want to think about an anti-infectives at this point, unless something changes in the landscape, commercial landscape with anti-infectives. Yeah. But again, we do support programs. If someone comes up and say, yeah, we have programs where we're supporting, guys in fungal group as well, your group, right? So we do support. In fact, we have a PhD student from the fungal group in our lab now, trying to get winners against their fungal target. So we support, but it's not something we tend to commercialize. Yeah.
Right. But I mean, it's great that as you said, that you're a non-immunologist, but we seem to have learned a lot more immunology. Like how B cells work in sharks, that sort of thing, which is often something we don't think about.
Yeah. It's quite fascinating actually. So the guys that made this discovery, actually, that was in 1993. It was just serendipity. They were looking at something else and they started seeing this weird thing, right? So it's quite strange how the immune system of shark, it's a weird one. It's a weird one that never really evolved, to be totally different. So, but yeah, it's quite fascinating. You have all the IG immunoglobulin molecules that you find in humans again, but then you have all these weird other ones that do hover around. But it's not, you have those guys who do shark immunology. We don't bother about that. It's not, we just tend to bother about the side of drug discovery, right? And let the smart guys do the immunology and tell us what they think. Yeah.
Yeah, well, I don't know about that, but yeah, it's been really fascinating. I mean, I think I wish you all the best. I think you're probably on the right track, and I think maybe we should all reach out more and to learn, because I mean, obviously, some of these technologies do not exist in the context where we work, and you're obviously a million miles away. And but yeah, I think more collaborations and more thinking about, because obviously Africa focus is always on infection, and that's what there is, right? Not that the other stuff doesn't exist, but yeah.
I agree. I see. Yeah. No, as always, I'm open, to support in any way which I can, as long as it's, something I can do. Absolutely, yes. Yeah.
And something that makes business sense.
As always, Sabelo.
Yeah. Thank you, Obinna. Thank you very much.
Thank you so much.
We very much appreciate.
Thank you for having me. I appreciate that as well. Okay, bye.
Cheers. Question and if you
the interesting thing is, yes, I agree. It's something that we all do, right? It's sometimes you don't want to ask a question because you might think that's stupid, right? No question is especially what we do, no question is stupid. And I find this with students. Most times I finish teaching and I try to implore them to actually go like one-on-one. Do you want to say anything? You find these ones that just wait for you outside.
Yeah.
The whole way and say, excuse me, can I ask you? But then guess what? They ask the best questions. And then you go, the answer would have benefited the entire class.
The class, yeah.
You know,
Why?
yeah.
It's a strange one. It's a thing that we need to unlearn,
Yeah,
I think.
absolutely. But someone is saying Frida wants to ask a question.
Yeah, go ahead.
Thank you very much. My question is about the use of antibodies to combat HIV. I know it might be a little bit a different topic, but I just want to get to pick your brains on this. So I was looking at including the code for anti-integrase antibody in the actual provirus of HIV. And one of the stumbling block in this theory of mine is the size of the actual code of the antibody against the integrase enzyme or the reverse transcriptase enzyme. But I have, when COVID started, I have read somewhere about a monomer or a relatively smaller antibody that was derived from camels. And I thought that if we could incorporate genetically that in the provirus of HIV, we may produce a virus that neutralizes other viruses. And my idea was on the line of having an HIV virus that have every gene or protein or the actual HIV virus, but put an addition in the region of the code for integrase, put a code for anti-integrase. In the region for a protease, put a reverse transcriptase, put a code for anti-reverse transcriptase there, so that when you have this virus integrated, then you produce viruses that are able to infect actually infect a host cell, but cannot integrate its genome into the host. But what they do is, if there is a co-infecting HIV within that cytoplasm, the proteases and integrases that came with this one will then neutralize and prevent infection. So to it, eventually you achieve clearance, because we know that persistence only survive on reinfection. And once you can achieve prevention of a reinfection, even the proviruses tend to expire as time goes on. Then you achieve clearance. So I just need an advice on that. And my focus is actually on my challenge is actually on the size of, and another thing is, if we include the size or that a code within the gene, will it, because I know HIV to be a big virus, could that hamper any of this idea? Could it maybe work? Could it maybe find ways that we can make it?
Very, very good question. So you're right. The first stumbling block of you trying to put a whole antibody into the genetic code there is it's complicated. So the thing is, so for you to actually remember, when I talked about the classical monoclonal antibody, we are talking about 4 chains that have to spontaneously come together. Okay, so you have 2 light chains, you have 2 heavy chains. So that's a very tricky one. Okay, so you have to have 2 vectors, not 1. So to solve that problem, it is actually achievable, but you need to have two vectors. One of the vectors making the heavy chain and one of the vectors making the light chain. But the challenge you then have is that the pairing has to then happen, in situ, you see? So which means that chances are you being able to neutralize your integrase may not work because the pairing will have to happen once the expression is up. You know what I mean? It happened for these different light chains. So the chains are not active unless they have paired together. Okay, so that's a big challenge. You're right to be looking into using a single domain, okay, because using a single domain, you evade that, you avoid that pairing, problem. And again, the size, it doesn't really matter so much about the size because your transfection is going to happen in human mammalian cells as depending on wherever you are targeting it, because you need to target to a certain cells for the expression to happen, right? So the size is not actually the problem. The only problem we see here is the pairing that would happen. So for a single smaller domain is an ideal way to go. And the burden, whether you have an anti-integrase and you have an anti-reverse transcriptase in one plasmid, that is not actually a big problem per se, because if you look at the expression vectors, these expression vectors do make a lot of other proteins. You have the chaperones, you have the, it do make a lot of other. So it's a technology that's been there. I don't know how effective that would be. I can't comment on that in terms of the actual efficacy, but I can, I'm commenting on you being able to design that. It's actually more plausible to do that with a single domain. Absolutely.
I don't know, can I ask a follow-up just a little bit? Yeah.
So can you just get one question from Dodo and then we come back to you? Is that okay?
Thank you very much.
Dodo, would you like to ask a question? So I think he was just summarizing on the chat what is the advantage of using VNAs over aptamers. I think that's the gist of the question.
Yeah, there are advantages. I think aptamers, you refer the single-stranded DNA molecule. Yeah, those are just short-term nucleic acid. When you say advantages, I won't call it like advantages. There are two different technologies. They are still small technologies, but you could argue that. So one thing you could argue is that you may be able to engineer in some. So if you could easily engineer, it's easier if you're working with a nucleic acid level molecular target, right? Molecular domain, in the sense that you can engineer in some features you want, right? With the VNA, it is not at that level. We need to express, it is a protein that we need to express, right? So this protein, for us to then engineer in features that we want, say we want to improve stability, for instance, say we want to improve stability against the pepsin, for instance, we need to go back and re-engineer this at a molecular, at a DNA level by improving the libraries, do lots of DNA mutation, at the library level, which is the DNA level, before we can then express again, do a transient expression, and then look at the features. So there are two different technologies, so you can't actually directly compare them. If you want to, the best comparison would be comparing the nanobodies, for instance. The nanobodies that come from camel will be a better comparison to what the VNAs do. Yeah.
Do we have time to take the last question? Do you want to come for that question again, Freddie? Obinna, sorry, do you have time?
No, it's fine. I mean, I'm surprised that the engineer hasn't. I hope they haven't killed him in my lab, but yeah, I've been looking. He hasn't come to my door, so it's fine.
Okay.
Yeah, I think it will rather be a very long one. I would rather ask privately, if possible. Maybe, I don't know how, because I was actually looking at I was actually intending to do this within the genome of HIV itself, not via plasmids.
Oh,
But
yeah. Okay. I see
yeah.
your point. Yeah, honestly, feel free to, I mean, again, it's not my specialty. I can only advise you on what, but feel free to contact me, whether LinkedIn, or just my name will lead you to my LinkedIn, actually. And absolutely, I will be happy to help and also direct you to an expert if it's out with my specialty.
Thank you very much.
Yeah.
Cool. Thank you, Freddie. And thank you all. Thank you. I'm assuming that we don't have any more questions. I do not see any. And yeah, thank you, Obinna. And thank you, everyone. Really appreciate the time you've given us. And yeah.
All right.
Cheers.